Hello everyone, I am Faizal Rahman. Today I will be talking about the topic using foundation model for gene regulatory network inference. Start off, gene regulatory networks or GRNs control the regulatory interactions between transcription factors and genes that drive cell differentiation and state transitions. Accurate GRN inference is crucial for understanding cellular functions. State-of-the-art computational methods use single-cell gene expression data for this purpose. On the other hand, foundation models pre-trained on large corpora of data have demonstrated the ability to excel in various downstream tasks pertaining to natural language and computer vision. Researchers have also developed foundation models specifically for biological domains, and these biological foundation models have shown impressive capabilities across different tasks, such as protein function and structure prediction. However, their general applicability in inferring GRNs remains uncertain. While state of the art computational methods leverage single cell gene expression data to predict GRNs, the application of foundation models in this domain remains largely unexplored. Thus, we aim to establish a robust framework to evaluate the potential of foundation models, such as SCGPT, in generating gene embeddings for GRN inference. Beeline framework is a well-established benchmarking pipeline to evaluate the performance of GRN inference methods. Sorry. It comprises standardized data preprocessing, containerized execution of GRN inference algorithms using Docker, parameter estimation, and evaluation. Beeline assesses the algorithms based on accuracy, stability, network motif analysis, and scalability. Inspired by the advances in GRN inference evaluation, and foundation models, we seek to establish a standardized framework for assessing the performance of foundation models in generating embeddings that are useful in inferring GRNs. In this study, we used SCGPT. In this study, we used SCGPT, a transformer-based model pre-trained on scRNA sequence data from 33 million human cells to compute gene embeddings from single-cell gene expression datasets. We then provided these generated embeddings as input to three GRN inference algorithms, namely Genie3, GRN Boost 2, and PIDC to predict GRNs. We used Beeline to predict the GRNs using the three methods and compare the performance when the raw gene expression data was provided as input to the same three methods. The data sets that we used, they have three types of ground truth GRNs: non-specific chipset, cell type specific chipset, and functional interaction networks. The two types of human single-cell gene expression datasets include human mature hepatocytes and human embryonic stem cells, that is HESC. The mouse datasets are as follows: mouse dendritic cells, mouse embryonic stem cells, and the three types of mouse hematopoietic cells. The results, of course, what we've been waiting for. So far, yes, the evaluation was done using two metrics: AUROC, that is area under receiver operating characteristic curve, and AUPRC, area under precision recall curve. The AUROC and AUPRC scores were calculated for the GRNs inferred using gene expression data from human mature hepatocyte cells. This provides a measure of how well the inferred GRNs capture the known regulatory relationships in this specific cell type. Additionally, the study looked at AUROC scores for GRNs inferred using seven different gene expression datasets and evaluated them against the non-specific chipset ground truth network. This gives a broader sense of performance of the GRN inference algorithms across multiple datasets. Interestingly, here we see the results show that for the mouse gene expression datasets, GRNs predicted using gene expression embeddings outperformed the GRNs predicted using the raw gene expression data in 33 out of the 45 experiments that we performed. This suggesting that the use of gene expression embeddings can improve the performance of GRN inference methods. As far as the summary and the future work is concerned, the study implemented a standardized approach using Beeline framework to evaluate the effectiveness of single-cell foundation models for gene regulatory network inference. The results show that the embeddings generated by the SCGPT single-cell foundation model outperformed the use of raw gene expression data in GRN inference. This was demonstrated across the GRN inference algorithms Genie3, GRN Boost 2, and PIDC. 7 gene expression datasets also, and 3 types of ground truth networks, that is, string, non-specific chipset, and cell-specific chipset. For future work, building on these promising results, we plan to expand the evaluation pipelines to include more single-cell foundation models such as SCBERT and GeneFormer. Additionally, we intend to explore the use of supervised fine-tuning techniques to adapt the single-cell foundation models to specifically for the task of GRN inference. This could potentially unlock even greater performance improvements by tailoring the embeddings to the unique characteristics and requirements of this downstream application. Thank you.